When I first heard the term gene editing, I imagined scientists in white coats, in sterile, white, futuristic labs, poring over petri dishes and microscopes. Little did I know that the first instances of gene editing occurred some 40 to 50 years ago, before I was even born. In fact, two biochemists in 2020 received the Nobel Prize for their work in describing how CRISPR works, which is a gene editing tool that allows us to make precise changes to the genetic code of an organism. This was predominantly due to the Human Genome Project, which started in 1990 and concluded in the early 2000s, with the sole aim of sequencing 3 billion base pairs of human DNA. Thus, although it may seem like a futuristic fantasy, it is already beginning to become an established technique. Already, gene editing has an important legacy in human history. For example, in the mid-1970s, scientists began to consider using recombinant DNA technology to produce human insulin from bacterial cells. The process involved taking a plasmid, a circle of bacterial DNA, if you will, and then inserting insulin into it, and reintroducing this into bacteria, which would be cultured in large vats of broth to produce pure human insulin that could be given to patients. This was much better than the synthetic version that had been used from pigs previously. Thus, we can see that although it may seem to be somewhat far-reaching, and in the future, it already is ever present, leading to the improvements of lives of many that suffer from the disease today. We have also begun to find initial treatments to cancer and Alzheimer's due to the identification of genetic mutations that cause such ailments. For example, recently, in clinical trials, researchers demonstrated the safety of using CRISPR to combat cancer. The process involved removing immune cells from 3 cancer patients and making genetic changes to improve anti-tumor immunity. And when these cells were reintroduced, they were well tolerated. Although they at best kept the cancer stable, they paved the way in the future for genetic CRISPR-engineered immunotherapies, potentially leading to a cure for cancer. Thus, although the legacy of gene editing is already being written, it has the potential to change the world as we know it. For example, by combating cancer, gene editing and engineering allows us to form quick diagnoses, effective interventions, be they changes in medicine, lifestyle, or even simple changes in diet. Furthermore, gene editing also has an important legacy for agriculture. With the ever-pervasive effects of climate change, by promoting drought resistance, we may produce more food to feed a growing population. With the population expected to reach 9.7 billion by 2050, this is a crucial development. Gene editing can lead us to produce higher food and greater quality produce by promoting resistance to droughts, pests, harsh weather conditions, and diseases. Perhaps you've heard of golden rice, where vitamin A is promoted and is used in developing countries, especially for children who may lack it. This has led to less deficiency and malnourishment in those developing countries. I would like to leave you with this. The potential of this medical panacea is endless, allowing us to cure potentially malaria, cancer, and vision disorders by making the lives of millions of people better as we change the genetic code very precisely. With the advent of superbugs and antimicrobial resistance, Just know that in future, when we or our children get sick, it may just be gene editing that saves us. It may just be those scientists in white coats poring over Petri dishes and microscopes that save us. It may just be CRISPR and the Human Genome Project that saves us. With fewer and fewer medicines keeping those pathogens at bay, just know that gene editing is potentially the best improvement in science so far. Thank you.